Shubhadeep sir, I am audible.
Okay.
Okay. Good morning, everybody. I am Amit Kumar Jana, state-aided college teacher, Department of Nutrition, Debra, Thana, Mahavidyalaya. Today, 31st July 2021, I welcome all of you in this national level webinar on nutrition and health, organized by Department of Nutrition and IQAC of Debra, Thana, Mahavidyalaya. Shubhadeep sir, I am audible.
একদম ক্লিয়ার।
Uh, we are thankful to our we are thankful to our principal ma'am and IQAC. I am Amit Kumar Jana and faculty members, teacher, Department of Nutrition, Debra, Thana, Today, 31st July 2021, I welcome all of you in this national level webinar on nutrition and health. Organized by I request all audience to keep your microphone muted.
একদম ক্লিয়ার।
We are thankful to our principal ma'am and IQAC. I am Amit Kumar Jana and faculty members, teacher, Department of Nutrition, Debra, there is some problem, network issue. I request for audience to give your microphone.
অমিত স্যার মনে হয় ওখানে ইউটিউব বা কিছু একটা খোলা আছে ওখানে, এখানে আওয়াজটা আসছে।
Sorry. Today, 31st July 2021, I welcome all of you in this national level webinar on nutrition and health, organized by Department of Nutrition and IQAC of Debra, Thana, Mahavidyalaya. We are thankful to our principal ma'am and IQAC and faculty members, Department of Nutrition, and also thankful to the technical team of our college with their cooperation and continuous support. This is impossible to organize to this webinar. We would like to thank our resource person who will make this webinar successfully by giving their valuable speech. Now, I would like to request our principal ma'am and chairperson of this webinar, Dr. Rupa Das Gupta, to start with inaugural speech. Over to you, ma'am. Ma'am.
Good morning. Am I audible?
Yes,
Yes.
ma'am.
Yes, ma'am.
শুভজিৎ শোনা যাচ্ছে?
ইয়েস ম্যাম। Yes ma'am, you are audible.
শোনা যাচ্ছে কি?
Yeah, yes
Yes,
ma'am.
ma'am, audible.
Yes, ma'am.
শোনা যাচ্ছে ম্যাডাম।
শুভজিৎ আমি কি শোনা যাচ্ছে কি আমাকে?
একদম শোনা যাচ্ছে ম্যাডাম, আপনি বলুন।
শোনা যাচ্ছে কি?
ম্যাডাম আপনাকে শোনা যাচ্ছে, আপনি বলুন, অসুবিধা নেই।
হ্যাঁ ম্যাম, শোনা যাচ্ছে।
ঠিক আছে, আমার স্পিকারে প্রবলেম হচ্ছিল।
আচ্ছা।
Sorry for this technical glitch. Well, good morning to all of you. What I can see is Dr. Sukanto Mondol has joined and probably Dr. Devidas Ghosh has joined. Amit Devidas sir ki join করেছেন?
Yes, ma'am. Yes, ma'am. Sajjal with us.
Okay, so good morning to the eminent speakers, Dr. Devidas Ghosh, Dr. Sukanto Mondol, to all the teachers of my college, and also to the participants, that the students of the Department of Nutrition. On behalf of Debra Thana Shohid Khudiram Smriti Mahavidyalaya, as a principal of this college, I heartily welcome Dr. Devidas Ghosh, Professor, Department of Biomedical Science, Biomedical Laboratory Science and Management, Vidyasagar University, and also to the other eminent speaker, Dr. Sukanto Mondol, Principal Scientist of Animal Physiology Division, ICAR, National Institute of Animal Nutrition and Physiology, Bangalore. Well, I won't be wasting my time because two eminent speakers will be speaking after me. Our college is a comparatively young college, and the science departments are younger, and we are trying our best to strengthen the infrastructure of the department in recent years. Some very fresh teachers have joined us SSCT in recent years, and they are also trying their bit to encourage our students to be not only to study the syllabus, to getting interested in the subject, to study, having a sort of inclination towards the subject. An organization of this webinar is a part of their endeavor in making our students aware of the things that are happening in their own subject, in their own discipline, and also interdisciplinary approach should always be there. No words are enough to thank Dr. Devidas Ghosh, because I won't be thanking him for joining this seminar, as Dr. Ghosh has always supported in whatever academic endeavors we have taken in. We have always got his support and encouragement. I thank Dr. Devidas Ghosh once again for his support in organizing this webinar. I have spoken to Dr. Sukanto Mondol earlier. I'm very thankful. I express my gratitude on behalf of the entire organizing committee of this webinar for his valuable time. I am sure that both of you, both our eminent speakers today, will enlighten our students and also us by their speeches. I once again thank and welcome all of you. Please continue with the webinar.
Thank you, ma'am, for inaugurating this webinar. This webinar would not have been possible without your constant support and guidance. Thank you, ma'am. Now, I like to request Pallavi Maiti Madam, faculty member, Department of Nutrition, and also Joint Organizing Secretary of this webinar, to say something about this webinar, Nutrition and Health. Over to you, ma'am.
Am I audible?
Yes, ma'am, audible.
Okay. Thank you, Amit sir. I'm really honored and grateful for giving me such opportunity to say something about the webinar. Honored distinguished person, precious academicians, our principal madam, our resource persons and researchers, scholars, all listeners, and my colleagues, and also my beloved students, a warm welcome to this webinar, Nutrition and Health 2021, organized by Department of Nutrition and IQAC jointly at Debra, Thana, Shohid Khudiram Smriti Mahavidyalaya. I also thankful to our technical team for their massive support. Actually, they are the backbone of this webinar. Without them, it is not possible. So we organize the webinar through Google Meet platform because currently the pandemic COVID-19 situation. The topic, the seminar focus on the health and nutrition is a broad topic. So as per the WHO, health is a state of complete physical, mental and social well-being, and not merely the absence of disease and infirmity. Nutrition is a critical part of health and development. Good nutrition means maintaining a nutritional status that enables us to grow well and enjoy good health. Nutrition and health is an amalgamation of science and healthcare, as is essentially the rest of the broad field of medicine. Besides this, nutrition play an important role to manage several medical conditions. Some of the health conditions that can benefit from changes enacted through the dietary modification include heart disease, diabetes, hypoglycemia, irritable bowel disorder, and obesity, high blood pressure, etc. Better nutrition is related to improve infant, child, and maternal health also, and stronger our immunity system, safer pregnancy and childbirth, and lower the risk of our life. Besides this, malnutrition in every form presents significant threat to human health today. The world faces a double burden of malnutrition that includes both undernutrition and also overnutrition. In this pandemic situation, nutrition is most necessary part of our daily life. Immune boosting diet should be include our daily diet, rich in vitamins and minerals, especially vitamin C, vitamin D, zinc, etc. Besides this, it is very important to all of us maintain sanitation and hygiene, use of sanitizer, maintain social distance, properly wear a mask, and stay happy, healthy, and stay at home. Now we are eagerly waiting for our next interesting and informative session to wise persons and our resource persons have with us. So I hope we all enrich ourselves from them. And so let's go ahead and get started our next session without wasting time. Over to you, Amit sir. Thank you, all of you.
Thank you, ma'am. Our inaugural session end here. Now we are going to our next technical session. Our resource person, Professor Devidas Ghosh sir and Dr. Sukanta Mondol sir are virtually present with us. I express my gratitude and gratefulness to our resource persons. Now I would like to request Snigdha Shah Madam, faculty member of Nutrition Department, also joint organizing secretary of this webinar, to welcome Professor Devidas Ghosh sir. Over to you, Snigdha ma'am.
Good morning. Good morning to all. Welcome to today's session. My respected principal ma'am, my respected resource person, my colleagues, and my beloved students. Let's be delighted to introduce Professor Devidas Ghosh sir, Professor and UGC Research Awardee, Department Biomedical Laboratory Science and Management, Vikas University. He was lecturer in Paspuro Bonomali College, Vikas University, senior lecturer, Paspuro Bonomali College, Vikas University, senior lecturer, Vikas University, reader and HOD Department of Physiology, Vikas University, professor, Vikas University from 2008 till now. His contribution in sciences, his research areas are endocrinology, molecular medicine, reproductive physiology, clinical nutrition, and nutrigenomics. List of PhD students under the guidance of Sir is 41. He has published different research papers in national and international level. National level 26 and international level 145. List of book publications are 4. Sir has already 2 Indian patents. His research work in India and abroad also. Research experience of Devidas Ghosh sir is 30 years. Another achievement of Professor Devidas Ghosh sir that Sir hold first position in Vikas University according to world ranking of scientists. Sir will talk on nutrition and health. Mainly, nutrients act as immunity booster during COVID-19 situation. Sir, please give your valuable speech.
Devidas sir, you can start. Sir, are you want to screen presentation?
Yes.
Okay, the option is
Please wait for 1 or 2 minutes.
okay. Now you can do.
Hello, hello.
Yes, yes,
দেখা
yes,
যাচ্ছে?
sir.
হ্যালো,
হ্যাঁ, 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 yes, sir, একদম।
দেখা যাচ্ছে? স্লাইড
হ্যাঁ, হ্যাঁ, দেখা
দেখা
যাচ্ছে।
যাচ্ছে তো?
হুম, হুম।
Okay. So, good morning and welcome all of you to this webinar. And I also offering my heartiest thanks and congratulations to the principal and the organizing team of this national webinar. I am also offering my heartiest thanks to another speaker, Dr. Sukanta Mondol, National Institute of Animal Nutrition, Bangalore. I like to speak both in English and Bengali because most of our students are also belong to from UG level. So for their understanding, I like to present my seminar as far as practicable in Bengali also. But sometimes I also like to speak in English, which are not actually can able to translate into Bengali properly. So today I like to focus that immunomodulator nutrients and COVID-19. Immunomodulator nutrients and COVID-19. And the title of this webinar, that is the nutrition and health, that is the most appropriate at this pandemic situation. And you know that we are actually in second wave of the COVID-19, and at any moment the third wave of COVID-19 may appear in our state also. And in that context, the role of nutrients is most important for immunoboosting, for immunoenhancement, for immunoenrichment, for immunoempowerment. Now, this COVID-19, that is the SARS-CoV-2 virus, if you like to study the SARS-CoV-2 virus, it is envelope virus having diameter 120 nanometer, and there are so many glycoprotein spikes which are crown-like virus and the single-strand positive sense RNA containing virus. The coming strain, that is the Delta strain of the corona, third wave, the Delta strain, where the spread, rate of the spread is very high, In case of the, you know that the alpha and the beta variant, the spreading rate is about 2 to 3. But in case of the Delta strain, the rate of the spread is about 9 to 10. So it is about 4 to 5 times more than the alpha and beta variant. And in that situation, most of the epidemiologists also suggest to improve our nutrition status so that we can able to increase our immune level. We can able to empower our immune activity. And thereby we can able to achieve the herd immunity. That is the population immunity. That is the community immunity. That is the people immunity. And from mathematical projection, it has been interpreted that to fight against that COVID-19, that herd immunity is also most important in this aspect. And about 78 to 80%, if the community members are immune against COVID-19, then the herd immunity would be achieved. Herd immunity would be achieved. Till now, we are not attained that level. Actually, serological survey is going on in our state. Serological survey. That means the antibody against the SARS-CoV-2 virus antigen. That study is also going on. And the headquarters of this serological survey study against COVID-19 in our state is School of Tropical Medicine, where one of my PhD students is the coordinator of that project at present. And from the yesterday data that my student also submit, that also convey that message, that in our state, the immunity, there is a herd immunity that we achieved about 61%. So we are far away from that 80% target level, 61% against that COVID-19. 61%. 61% of the individuals in our community has the antibody against that SARS-CoV-2 antigen. And for that reason, most of the SARS-CoV-2 beta variant and alpha variant are asymptomatic. So in that present situation, the most of the experts in that COVID-19, their suggestion is that try to improve immunity by food style modification, by lifestyle modification, along with vaccination program. But we know that we are far away from the target of the vaccination covering. In our state, about 14% vaccination has been completed of the total population. 14. We are far away from that 100%. So the alternative way is to raise that herd immunity by immunoboosting nutrients, as well as by social engineering. That is by masking, by sanitation, and by others. Okay. So regarding the introduction, I like to say that is a nutrition and health, where health means well-being of physical, social, mental, emotional, psychological health, etc. But actually, the health means adjust or adapt the environment. When an individual is unable to adjust or adapt the altered environment, then we may say that his or her health is not good. Similarly, in that pandemic situation, when we are unable to adapt ourselves to that environment, that means when we are unable to fight against that SARS-CoV-2 infection, then our health is not okay. Our health level is not up to the standard. And that health status has been assessed against COVID-19 infection from the viewpoint of immunity, which is known as immune health. So immune health at present is the most important part of that pandemic situation. And that immune health may be modulated drastically in positive direction by food style modification and by lifestyle modification. Both the undernutrition and overnutrition, আমরা জানি যে আমাদের যদি নিউট্রিশন কম হয় বা ওভার নিউট্রিশন হয়, দুটোই আমাদের হেলথ ডিসঅর্ডারস ঘটায়। And for this reason, so many diseases are at present known as food style diseases, lifestyle diseases, like diabetes, CVD, এগুলোকে আগে বলা হতো metabolic disease. We also changed their name, that these are known as the lifestyle, food style disorders. So that nomenclature actually focus that what is the importance of the food style, what is the importance of the lifestyle for health maintenance and for disease prevention. Now we like to focus today that is the nutrients for immunomodulation, that is a positive immunomodulation, not negative. That is the immuno enhancement, immuno enrichment, immune empowerment, not immuno weakening. That is not negative immunomodulation. And that nutrients, maybe vitamins, maybe some nutraceuticals, may have some minerals also, can able to modulate in positive direction both the cellular as well as the humoral immunity, which are the two wings of our immune system. And when that health status is deteriorated, then the disease is also noted. And out of these, that the communicable disease, we are like to focus today that the communicable diseases. That means the diseases are transmitted by several microbes, by several agents, by several microbial agents. And that communicable diseases are divided into two types, infectious and contagious. Sometimes we say that infectious or contagious, but actually infectious and contagious are not identical. All contagious diseases are infectious, but all infectious diseases are not contagious. যেমন আমরা বলছি ইনফেকশাস ডিজিজ কি, যেটা হচ্ছে মাইক্রোবায়াল এজেন্ট দ্বারা হচ্ছে, বাট ইটস স্প্রেডিং, it is not spread from one to other individuals. As for tetanus, tetanus is infectious disease. Mycobacterium tetani, you know that is a specific microbial agent responsible for the onset of the disease. কিন্তু আমি যদি একটা টিটেনাস পেশেন্টের পাশে বসি, তার সঙ্গে যদি আমি টাচ করি, তার সঙ্গে যদি একসঙ্গে খাই, there is no chance for the infection, for the tetanus infection. So that is infectious, but not contagious. But in case of COVID-19, COVID-19 is also SARS-CoV-2 microbial agent. কিন্তু যদি আমরা COVID-19 পেশেন্টের পাশে বসে টাচ করি, there is a high chance of that infection. It is a contagious. So contagious are infectious, but all infectious diseases are not contagious. Am I clear? As for example, COVID-19 is infectious as well as contagious, but tetanus is only infectious, but not contagious. Okay? So today we also focus our lecture on that contagious disease, that is the COVID-19. And that COVID-19 third wave is inevitable. আমরা এটাকে বাধা দিতে পারবো না, আমরা এর থেকে মুক্ত হতে পারবো না। It is likely to hit India within 6 to 8 weeks. According to prediction of mathematical calculation, as well as by epidemiologists, they also predict that within coming 6 to 8 weeks, they also hit the India. Already some states are actually under third wave. And in this condition, human behavior, that is referred as the social engineering process, social engineering, mind that. Social engineering is the most important measures against that COVID-19. Like physical distancing, by which you can able to spread this disease, hand washing, regular hand washing, mask wearing, facial mask, and symptoms monitoring. That means if there are symptoms of that COVID-19, you also go to the diagnostic test and take the proper measures so that the spreading can be prevented. So these are the social engineering steps: physical distancing, hand washing, mask wearing, and symptoms monitoring. And if we follow these steps properly, then the high spread, super spreader Delta strain of that SARS-CoV-2, that virus can be protected. Otherwise, that spreading rate would be so high that we are unable to protect ourselves from that third wave of this COVID-19. Okay? Now, why immunomodulation is so important in our country? Why immunomodulation? In our country, first cause is that healthcare system is not well developed as US or other developed countries. Actually, healthcare system, আমাদের যে স্বাস্থ্য পরিষেবা ন্যাশনাল লেভেলে আমরা ধরে বলছি উন্নত নয়, যদিও আওয়ার স্টেট, আমাদের রাজ্য সরকার ট্রাই লেভেল বেস্ট সর্বোচ্চভাবে চেষ্টা করে চলেছে টু আপগ্রেড দ্যাট হেলথকেয়ার সিস্টেম অ্যাজ ফার অ্যাজ প্র্যাকটিক্যাবল, যতটা সম্ভব, টু ফাইট এগেন্স্ট দ্যাট COVID-19. তারা অনেক হসপিটাল এস্টাবলিশ করেছে, COVID-19 হসপিটাল, এবং সেগুলোর দ্বারা COVID-19 এগেন্স্টে ফাইট করে চলেছে। তথাপি আমাদের আরো ডেভেলপ করতে হয়। অ্যাকর্ডিং টু আমার স্টেট গভর্নমেন্ট স্টেটমেন্ট, তারা বলেই চলেছেন যে আমাদের আরো ডেভেলপ করতে হবে। তার জন্য আমরা বলছি হেলথ কেয়ার সিস্টেম এখনো আমরা একবারে সর্বোচ্চ লেভেলে আসতে পারি, তাহলে আমাদের সেলফ প্রোটেকশন দরকার। এবং সেই সেলফ প্রোটেকশনটা একটা ইমিউনোমডুলেশন। এবং এই হেলথ কেয়ার সিস্টেম আমরা যেটা বললাম ভ্যাকসিনেশন, দ্যাট ইজ অলসো ওয়ান টাইপ অফ পজিটিভ ইমিউনোমডুলেশন। কিন্তু আমরা ভ্যাকসিনেশনে খুব দূরে আছি, লক্ষ্যমাত্রা থেকে অনেক দূরে আছি, কারণ সেন্ট্রাল গভর্নমেন্ট আমাদের ঠিক মতো ভ্যাকসিন পাঠাচ্ছে না। এবং ভ্যাকসিন না পাঠানোর ফলে আমাদের রাজ্যে বিশেষ করে 18 to 45 এই গ্রুপটা খুব কম ভ্যাকসিনেশন পেয়েছে, ভেরি পোর ইন রেসপেক্ট টু দ্য আদার স্টেট। তাহলে আমাদের ইমিউনোমডুলেশন ক্ষেত্রে আমাদের ফুড স্টাইল মডিফিকেশন ছাড়া এখন আর কোনো উপায় নেই। তার সঙ্গে সোশ্যাল ইঞ্জিনিয়ারিং হয়েছে আগের স্টেপগুলো। 2 নম্বর হচ্ছে কেন আমাদের দেশে ইমিউনোমডুলেশন দরকার, হেভিলি ক্রাউডেড কান্ট্রি। So human behavior not able to follow in every time. আমরা সবসময় এই সোশ্যাল ইঞ্জিনিয়ারিং প্রসেসটা ফলো করতে পারবো না। সোশ্যাল ডিস্টেন্সিং, মাস্ক ওয়ারিং সবসময় আমরা পারি না। আমরা গ্যাদারিং করে ফেলি অনেক সময়। তাহলে আমাদের কি করতে হবে? আমাদের যদি ইমিউনো মডুলেশন ঠিক থাকে, আমরা যদি ইমিউনো এম্পাওয়ারমেন্ট, আমরা যদি হার্ড ইমিউনিটি গড়ে তুলতে পারি, তাহলে আমাদের এই ক্রাউডেড কান্ট্রিতেও আমরা আমাদের প্রোটেকশন নিতে পারবো। আর নেক্সট ওয়ান, ইকোনমিক কন্ডিশন ইজ নট গুড। আমাদের একটা কোভিড-19 যদি হয়ে যায়, একটা দেখেছো প্রাইভেট হসপিটাল খরচা কত, 8 থেকে 10 লাখ টাকা, আরো বেশি কিছু কিছু যায়। সেন্ট্রাল গভর্নমেন্ট, স্টেট গভর্নমেন্ট হসপিটালে ফ্রি অফ কস্ট, কিন্তু প্রাইভেট হসপিটাল সবাই তো আর মানে স্টেট গভর্নমেন্টের হসপিটালে যায় না। স্টেট গভর্নমেন্টের হসপিটালে ট্রিটমেন্ট বিনা পয়সা, কিন্তু অন্য হসপিটালে অনেক খরচা। তাহলে ইকোনমিক কন্ডিশন ইজ নট গুড। তাহলে আমরা যদি ইমিউনো মডুলেশন করে এটাকে প্রোটেকশন দিতে পারি, so it is the most important measure against that COVID-19. So for these three reasons, I like to say that immunomodulation, positive immunomodulation is the most important way or weapon at present, most important weapon to fight against that COVID-19. Now, this public health, that COVID-19 infection is also under public health, and for its protection, 3 P's are very much essential. There is a projection, controlling measure to be projected on the community on the basis of the risk zone. So projection is the most important way to fight against that. Next one, prevention. Prevention by social way, prevention by vaccination program, prevention by dietary approach, that is food style and lifestyle. And another one, the promotion, that the hygienic promotion, healthcare promotion, and the healthcare service promotion. So by projection, by prevention, and by promotion, this pandemic situation in our country may be tackled to some extent. Now, if we study the prevention pyramid, disease prevention pyramid, it has been revealed that there are 4 levels, 4 strata for this prevention. And first one is the primordial prevention. This is purely non-clinical part, primordial prevention, where lifestyle modification, food style modification, pregnancy nutrition, lactating nutrition, are the most important ways, important avenues for the primordial prevention against the disease. And in that strata, in that primordial prevention, in that strata, the nutritionist play an important role for such immune enhancement. Next one, primary prevention. It is also a non-clinical part where information, education, and communication, IC, is the most important. Through information, through education, and through communication, we can able to develop the knowledge of the community member against such pandemic condition. And when the knowledge is upgraded, then the attitude of the individual will be changed. Health-friendly attitude will be developed in that community members. And when that health-friendly attitude is developed, then health-friendly practices would be adopted by the community members. And when most of the community members follow that health-friendly habits, health-friendly practices, health-friendly behaviors, then we can able to fight against that COVID-19. So this is the primary prevention, primordial and primary, where both the nutrition, in both the strata, the nutritionists take an important role for such prevention. And secondary prevention, mainly there is the clinical aspect by different vaccination, by different medicine, etc., etc. And tertiary, that is also major contribution by clinical person, that is by treatment, that is by rehabilitation. So by these 4 strata, out of these 4 strata, primordial prevention and primary prevention, where the nutrition modification, lifestyle modification, these are most important aspects for immune enhancement and to fight against that COVID-19. Now, also not clear properly. And in this vaccination program, some of the research about that clinical trial phase 3, phase 4 is also going on. And further, as you know, that interval between the first dose and second dose is also changing. And this is the part of the research, which also indicates that we are also till now in research, though the vaccination is going on to save the human life. So actually, some rules have been break here. আমাদের কিছু নিয়ম ব্রেক করতে হয়েছে কোভিড-19 ভ্যাকসিনেশন ক্লিয়ার করার জন্য। আমরা ফেজ 3 ফেজ 4 ক্লিয়ার না করে এসেছি, কিন্তু আমাদের রুলস আগে না হিউম্যান লাইফ আগে। হিউম্যান লাইফ আগে বলেই রুলসকে আমরা ব্যতিক্রম করে আমাদের এই কোভিড-19 ভ্যাকসিনেশন প্রোগ্রাম আমরা দিতে শুরু করেছি, এবং এর ফলে কিছু কিছু অসুবিধা আসছে, সেগুলো আমাদের এখন ওভারকাম করার জন্য চেষ্টা করা হচ্ছে। So now this disease prevention, I have the first point as the immunoboosting to overcome the immunodeficiency, and that immunoboosting may be conducted by individual-centric diet therapy. Individual-centric diet therapy. Antioxidant and rich nutraceutical. Because you know that COVID-19 is also associated with oxidative stress. COVID-19 is oxidative stress. And that oxidative stress that generates free radicals, which are responsible for the destruction of T lymphocytes and B lymphocytes, which are the major component of the cellular and humoral immunity. So if we consume the antioxidant and rich nutraceuticals through our diet, তাহলে আমরা কি করতে পারি? এই কোভিড-19 আমাদের যে ফ্রি রেডিক্যালস জেনারেট হচ্ছে, সেই ফ্রি রেডিক্যালস জেনারেশনটাকে আমরা ট্যাকেল করতে পারি। And that antioxidant is nutraceutical by food style modification. Nutrient drug interaction is another aspect by which you can able to increase the therapeutic efficacy of the drug by the modulation of the nutrient. And the last step, the nutrigenomics. That the nutrient, I like to focus today that the most important vitamin, which is immunoboosting, which is the non-classical function of vitamin, that is the vitamin D. That the nutrigenomics. That this vitamin can able to modulate the gene expression of different macrophage cells, of the different helper T cells, and thereby they can able to modulate the cellular and humoral immunity. Okay. Now we like to focus that is the vitamin D. Vitamin D. আমরা ভিটামিন ডি জানি সবাই, এবং ভিটামিন ডি এর ফাংশন আমরা সবাই জানি। আমরা মেজর ফাংশনগুলো জানি, অস্টিওজেনেসিস, বোন ফর্মেশন, টিথ ফর্মেশন, কিন্তু এগুলো তো ক্লাসিক্যাল ফাংশন। নন ক্লাসিক্যাল ফাংশন যেটা মোস্ট ইম্পর্টেন্ট, আমাদের এই প্যান্ডেমিক সিচুয়েশন না এলে আমাদের এই ফাংশনটাই হয়তো আমাদের জানা থাকতো, যে ভিটামিন ডি ইমিউনোমডুলেটর, পজিটিভ ইমিউনোমডুলেটর। এবং সব থেকে ইম্পর্টেন্ট বিষয়বস্তু হলো আমাদের মতো এই পোর কান্ট্রি, ডেভেলপিং কান্ট্রি, যে ভিটামিন ডি প্রায় 70% অফ দ্য আরডিএ, আরডিএ কত? 600 microgram per 600 IU per day, 600 IU per day. If 70%, near about 400 to 450 IU, we can able to synthesize in our skin through UVB light. UVB light exposure আমরা নিজেরাই সিনথেসিস করতে পারি, কোনো কষ্ট লাগবে না। আর ভিটামিন ডি এর যদি ভ্যাকসিন, ট্যাবলেট কেন, ট্যাবলেট বলছি, ক্যাপসুল কেন? ভিটামিন ডি ক্যাপসুলের প্রচুর দাম। ভিটামিন ডি ক্যাপসুলের দাম অন্তত মিনিমাম একটা ট্যাবলেটের দাম 1000, 1200, 1500 টাকা অন দ্য বেসিস অফ দ্য ইউনিট। কিন্তু দেখো আমরা নিজেরাই এতে ভিটামিন ডিটা তৈরি করতে পারি উইদাউট এনি কস্ট, কিনে আমরা তৈরি করতে পারি। এবং সেটা কত না, নিয়ার অ্যাবাউট 70% অফ দ্য ডেইলি রিকোয়ারমেন্ট, 70% অফ দ্য ডেইলি রিকোয়ারমেন্ট। এবং এটা কখন হয়, যে দ্যাট ইজ দ্য সানলাইট এক্সপোজার, সান বাথ এটাকে বলে। এটাকে বলে হেলিওথেরাপি, হেলিওথেরাপি, হেলিও, HLIO, হেলিওথেরাপি, সানবাথ। এবং সেটা কখন? আমাদের আগে ধারণা ছিল সকাল 8টার সময় যদি আমরা 8টা 9টার সময় যদি সানবাথ করি তাহলে আমাদের ভিটামিন ডি তৈরি হবে। কিন্তু এখন রিসার্চে বলা হচ্ছে না, ইট ইজ টোটালি রং কনসেপ্ট, ইট ইজ টোটালি রং কনসেপ্ট। 10টা থেকে 3টে, সকাল 10টা থেকে 3টে, when the UVB light is maximum. UVB light, you know that is wavelength is about 280 to 320 nanometer. And এই UVB লাইটে আমাদের স্কিনে ভিটামিন ডি সবচেয়ে বেশি তৈরি হয় 10টা থেকে 3টে। এবং প্রতিদিন যদি আমরা 15 মিনিট, অনলি 15 মিনিটস টু 20 মিনিটস এক্সপোজার আমরা নিই, বেশি দরকার নেই, 15 থেকে 20 মিনিট, দ্যাট ইজ সাফিসিয়েন্ট ফর দ্যাট রিকুইজিট অ্যামাউন্ট অফ ভিটামিন ডি। প্রতিদিন 15 থেকে 20 মিনিট, সকাল 10টা থেকে 3টে। এবং এর ফলে আমাদের ইমিউনোমডুলেশন হয়। এই জায়গাটায় আমরা দেখাতে চাইছি হাউ ভিটামিন ডি ক্যান এবল টু হেল্প দ্য পজিটিভ ইমিউনোমডুলেশন। Now you also know that vitamin D যেটা আমরা খাই সেই ভিটামিন ডিটা কিন্তু বায়োঅ্যাক্টিভ নয়। It is not bioactive. It undergoes first oxidation in the liver, hydroxylation in the liver, hydroxylation. And then second hydroxylation is going on in the kidney, renal tissue, kidney. And as a result, the 125 dihydroxycholecalciferol or calcitriol is formed. And that calcitriol, this is known the biotransformation, bioactivation of vitamin D. And that biotransformation or bioactivation of vitamin D is very much essential for immunomodulation. পরে দেখা গেল না শুধুমাত্র কিডনি টিস্যু নয়, আগে আমাদের ধারণা ছিল লিভার ফার্স্ট হাইড্রক্সিলেশন and second site of hydroxylation is the renal tissue. But from research work it has been noted that macrophage cells, which are the most immune cell, important immune cell, can able to hydroxylate the 25 hydroxylated vitamin. That means the renal tissue that conduct the second hydroxylation is also conducted by macrophage cells. I like to focus it. Yes, this is the vitamin D liver, where it goes first hydroxylation and it's from 25 hydroxylated vitamin D. But the renal, besides the renal cells, immune cells like T cells, like B cells, like macrophage, like dendritic cells, that is antigen presenting cells, are also the site for the second hydroxylation of 25 hydroxyl vitamin D, and as a result 125 dihydroxylated vitamin D will be formed, 125 dihydroxycholecalciferol. Okay, so that focus that immune cells, why they can able to hydroxylate these 25 hydroxylated calciferol. Now these cells, after biotransformation through second hydroxylation, can able to form active vitamin D, and these are responsible for interaction of some genes in that macrophage cell, in that dendritic cell, in that T cell, and can able to synthesize some biomolecules that I like to focus later on. That is the human beta defensive protein 2, HBD2. So these are the most important biomolecules which have the capacity, which have the efficacy to fight against that SARS-CoV-2 virus or RNA virus or single-stranded positive sense RNA virus. Now, what is the potent effect of the vitamin D3? Vitamin D3 also against respiratory tract infection. There is a virus who infect the respiratory tract, like the SARS-CoV. It mainly affects not all type of virus. It is an antiviral agent, okay, but against the enveloped virus like SARS-CoV-2. Enveloped virus, যার বাইরে envelope রয়েছে, একটা কোটিং রয়েছে, একটা খোলক রয়েছে, তাদের against ভিটামিন ডি ভালো কাজ করে। And from nutrient-gene interaction, it has been indicated that vitamin D3 can able to produce two most important biomolecules through nutrient-gene interaction. One is known as cathelicidin. Cathelicidin is the first biomolecule, cathelicidin, most important biomolecule for enhancement of the cellular immunity and to some extent the humoral immunity. We like to focus later on that what are the ways, what are the processes by which cathelicidin can able to enhance the cellular immunity and the humoral immunity for positive immunomodulation. And another biomolecule that vitamin D can able to synthesize in the macrophage cells, that is human beta defensive 2 protein, HBD2. That HBD2 fight directly against that COVID-19, So these are the two most important weapons as biomolecules, which are synthesized in macrophage under the guidance of the vitamin D3. And from that mechanism of action, vitamin D3 at present now called as most important immunomodulator. So this is the slide about that positive immunomodulation about vitamin D3. So macrophage cell, suppose this is the macrophage cell, this is the central part of the macrophage cell. When COVID-19, that is the SARS-CoV-2 viral antigen, attacks the macrophage cell through antigen presenting cell, okay? It has two probability. It has two probability. That pro-inflammatory cytokine wing may be stimulated. Pro-inflammatory, at the left-hand side, pro-inflammatory cytokine wing may be stimulated by enhancing the T helper type 1 cells, TH1 cells, T helper 1 type cells. And another possibility is that if this left wing would be developed, would be stimulated, would be enhanced, would be activated when vitamin D is deficient in our cell. Vitamin D is deficient in our blood. And that right hand wing would be activated when vitamin D level is sufficient. When vitamin D level is sufficient. Okay? And normal condition, there is a homeostasis between this anti-inflammatory wing and pro-inflammatory wing. Anti-inflammatory wing and pro-inflammatory wing. So when vitamin D is deficient in our body, then under the guidance of this macrophage, helper T1 cells, you know that the T lymphocytes are also several types. Helper T lymphocytes, killer T lymphocytes, suppressor T lymphocytes, memory T lymphocytes. But I like to focus the helper T lymphocytes. Helper T lymphocytes are also different types. Helper 1, helper 2. And this helper 1, when stimulated, then it can able to release interleukin 1, interleukin 6, interleukin 17, 21, etc. And out of these, interleukin 6 is the most important, is the captain of the pro-inflammatory cytokine school. Pro-inflammatory cytokine school, the interleukin 6 is the captain. And when vitamin D is less than the normal level, and at this condition, if there is COVID-19 infection, there is the SARS-CoV-2 infection, then that pro-inflammatory cytokine levels are elevated in our blood, and that is known as the cytokine storm. So, cytokine storm, cytokine storm. High level of this interleukin, pro-inflammatory cytokines level are elevated. And these pro-inflammatory cytokines results the MOF, that is a multi-organ failure. Multi-organ failure, it may result lung failure, pulmonary failure, it may result renal failure, it may result cardiac failure, and ultimately the mortality is noted. But if the vitamin D is present, then this is shifted to the right side. And as a result, the macrophage in presence of vitamin D can able to stimulate the helper 2 lymphocytes. Helper 2, not helper 1. And due to the helper 2 lymphocytes, anti-inflammatory cytokines level will be increased. Like interleukin 4, interleukin 5, interleukin 10, etc. And due to such high level of the anti-inflammatory cytokines, when the titer of the anti-inflammatory cytokines is increased, then that multi-organ failure that would be decreased. And 7 to 10 days only required for the shifting from the right hand side to the left hand side. There is a pro-inflammatory side to anti-inflammatory side if the vitamin D is present. So this is the most important slide about the role of vitamin D at the immune cells. আমি সব স্লাইডগুলো যাচ্ছি না, তোমাদের সময় অত বেশি নেই হয়তো। আমি আরেকটা স্লাইড দেখাচ্ছি এখানে রিগার্ডিং দ্য role of vitamin D. Now this is the macrophage cell. This is the macrophage cell. This is the center. This is the macrophage cell. And that vitamin D, that is by second hydroxylation, it forms vitamin D, which can able to bind to the vitamin D receptor, VDR. And that VDR can able to transmit the signal to the gene. And by nutrient gene interaction within this macrophage, it can able to synthesize catalycidine. One biomolecule. Second biomolecule, that is known as a HBD, human beta defensive protein. And these are the switch on process. There is the upregulation of catalycidine gene and human beta defensive 2 gene under the guidance of vitamin D3 in macrophage. This is the upregulation. Upregulation, that is the switch on of the gene expression. Side by side, vitamin D in the macrophage can able to downregulate, that is the switch off of nuclear factor kappa B, which is a transcription factor, which is responsible for pro-inflammation, that is responsible for MOF. So as vitamin D can able to switch off this nuclear factor kappa B, which is the transcription factor, which is very much essential for the synthesis, for the gene expression of interleukin-6. So due to the low level of the NF kappa B, interleukin-6 synthesis would be decreased, and thereby we can able to protect ourselves, protect the patient who are suffering from the SARS-CoV-2 from MOF, that is the multi-organ failure. Now, what is the function of cathelicidine? There are three ways of the cathelicidine. First one, that is chemotaxis. You know that আমাদের ম্যাক্রোফাজ সেল, আমাদের নিউট্রোফিল, আমাদের মোনোসাইটস, এরা কোথায় যায় না, যেখানে আমাদের হচ্ছে মাইক্রোবায়াল এজেন্ট ইনফেক্ট করে যে টিস্যুগুলোকে, সেই টিস্যুর দিকে ছুটে যাওয়া, তাকে বলে chemotaxis. That is the migration of the phagocytic cells, migration of the defensive cells from the vascular bed towards the zone of infection. That migration is referred as chemotaxis. So cathelicidine can able to stimulate the chemotaxis process, and as a result, the immunoresponse will be activated. Side by side, this cathelicidine also increases the second way, that the vascular permeability of the polymorphonuclear leukocytes, PMNL, polymorphonuclear leukocytes. Vascular permeability, that is the capillary permeability, and that is known as the diapedesis. That is the most of the macrophage cells, most of the phagocytic cells can able to migrate from the capillary bed to the tissue space through the capillary wall, which is referred as diapedesis. And cathelicidine can able to improve that vascular permeability, and so the shifting of that phagocytic cells towards the zone of infection will be increased. যেমন আমাদের সমাজে কি হয়, যদি কোনো আমাদের মানে কি বলবো অ্যান্টি সোশ্যাল আক্রমণ করে, তখন আমাদের কি হয়, চারদিক থেকে পুলিশ এসে ওই জায়গাটা ঘিরে ফেলে। ঠিক সেরকম যেখানে ওই অ্যান্টি সোশ্যাল, মানে এখানে আমরা বলছি যে মাইক্রোবায়াল এজেন্ট কোনো টিস্যুকে অ্যাটাক করেছে, তাকে ধ্বংস করার জন্য চারদিক থেকে আমাদের এই পুলিশ মানে হচ্ছে ডিফেন্সিভ সেল, মানে নিউট্রোফিল, মোনোসাইটস চারদিকে আসতে হবে। যেটা ছিল কোথায়, ব্লাড থেকে ক্যাপিলারি ওয়াল ভেদ করে তারা ওই জায়গায় আসবে। এই আশাটাকে সাহায্য করছে ক্যাথলিসিডিন। আরেকটা ক্যাথলিসিডিন কি হেল্প করে না, এরা হচ্ছে টি লিম্ফোসাইটস, বি লিম্ফোসাইটস, তার পলিফারেশন, তার প্রসেসিং, তার অ্যাক্টিভেশন, তার ডিফারেন্সিয়েশন, that is the cathelicidine also control this process. এগুলো অনেক ডিটেলসে, এগুলো আমরা অত ভেতরে যাচ্ছি না। proliferation, activation, তার প্রসেসিং, the T lymphocytes that is responsible for cellular immunity, and the B lymphocytes that is responsible for humoral immunity. কেন? the B lymphocyte থেকে তৈরি আমাদের plasma cells, and that plasma cells are responsible for immunoglobulin synthesis, IgG, IgA, IgD, IgE, IgM. We also know it. So another biomolecule I like to focus on the human beta defensive 2. That human beta defensive 2 that directly তারা কার উপর কাজ করছে, ভাইরাসের এনভেলাপটার উপর কাজ করছে। ভাইরাস এনভেলাপের উপর কাজ করে কি করছে, এরা পোর্ট সৃষ্টি করছে, এনভেলাপটার উপর পোর্ট সৃষ্টি করছে। And due to the port there is water influx, প্রচুর জল ঢুকে যাবে ভাইরাসের সেলে, and the virus will be burst. The biomolecules of the virus then would be deteriorated. So membrane degradation would be noted due to such influx of water through the pores under the guidance of HBT, which is synthesized in macrophage cells under the supervision of vitamin D. And due to such membrane degradation, there is a question about the validity, sustainability of SARS-CoV-2. ভাইরাস থাকতেই পারছে না, এক্সিস্টেন্স করতে পারছে, এক্সিস্টেন্স না করতে পারলে তারা প্রলিফারেট করতে পারবে না, এবং তার ফলে আমরা আমাদের ওখান থেকে প্রোটেকশন পেয়ে যাবো। তো দিস আর দ্য মোর স্পেসিফিক রোল অফ দ্য ভিটামিন D3. আমি জাস্ট বেশি বলছি না, অলসো দিস ইজ দ্য পেপার অ্যাবাউট দ্য cathelicidine and vitamin D3. This is the immunology about that vitamin D and cellular and humoral immunity. This is another paper that the vitamin D supplement may prevent the millions of winter infection, that is a viral infection. Okay. Now, similarly the zinc, আমি একটা ছোট করে আসছি আর বেশি বলছি না, zinc is also another immunomodulator. Zinc has antiviral activity, you also know it. because the gene can able to inhibit the binding of the SARS-CoV-2 virus with that angiotensin converting enzyme receptor which are present along the pulmonary alveoli. Pulmonary alveoli যে AC receptor যেখানে bind করে কে SARS-CoV-2 ওই বাইন্ডিংটাকে ইনহিবিট করে দেয় জিন। It also interferes the viral uncoating. Viral কে uncoat করতে দেয় না, মানে envelope থেকে বেরোতেই দেয় না, envelope থেকে ভাইরাসকে বেরোতেই দেয়। It also prevents the viral transcription and translation process. Transcription and translation processটাকে inhibit করে। It also inhibits the proteases and polymerase enzymatic process of virus. Polymerase enzyme এবং তার ফলে তার RNA replication করতে পারে না। So that gene is the most important modulator, immunomodulator, positive immunomodulator. And this gene also binds with a particular protein and the zinc finger protein, and it inhibit, আমরা আগেই বলেছিলাম যেমন ভাবে আমাদের ভিটামিন ডি করে এও ইনহিবিট করে। Zinc also have a nutrigenomic effect. It also modulates the gene. Zinc also modulates the gene expression of nuclear factor kappa B, transcription factor activation. এবং এর ফলে আমাদের কি হয় না, pro-inflammation would be decreased, anti-inflammation would be elevated. And side by side, zinc also the most important cofactor of superoxide dismutase. It's the antioxidant enzyme, endogenous antioxidant enzyme, and that can able to overcome the oxidative stress. And by that joint action, the zinc is also most important positive immunomodulator. Vitamin C, more than 20 different ways it can able to exert its activity for positive immunomodulation. It also increases the production of the T and B lymphocytes, most important. Next one, vitamin C also enhances the gaseous hormone synthesis in the endothelial cells of the blood vessels. There is the nitrogen oxide. It enhances the production of nitrogen oxide. Nitrogen oxide is the humoral. There is the gaseous hormone, only gaseous hormone which is present in our body. Sorry. হ্যালো মাদি, হ্যাঁ মেলটা যায়নি, আমি দেখেছি ফেল করেছি, আমি আজকে পাঠাচ্ছি। আমি একটা সেমিনার আছে, পরে একটু কথা বলছি। হ্যাঁ ঠিক আছে, হ্যাঁ hello, sorry. T and B lymphocyte production is also enhanced, and it also inhibits the production of neuraminidase. Neuraminidase is the most important enzyme for the viral proliferation in host cell. Host cell viral replication করবে, viral proliferation করবে, তার জন্য neuraminidase enzyme is very much essential. এটা কি করে? Vitamin C can able to inhibit this neuraminidase, and thereby the viral replication cycle in that host cell will be prevented. So there are so many ways of the vitamin C. The vitamin C is also immunomodulator. And what are the requirement dose? This is the RDA. তোমরা জানো বিভিন্ন বই পড়ে পেয়ে যাবে ভিটামিন ডি এর আমরা বলেছিলাম যে the 600 IU per day, 600 IU. And the vitamin C about on the basis of the age group, it is also RDA is also changed. Though in 2020, next 2020 যে the latest modification of ICMR, সেখানে দেখবে RDA টা একটু চেঞ্জ হয়ে গেছে। নাও আমরা যেটা বলছি এটা আমাদের একটু জেনে রাখা দরকার যে আমাদের ডায়াবেটিস এবং কোভিড-19 ইনফেকশন, ডায়াবেটিস এবং কোভিড-19 ইনফেকশন এটা খুব কোমরবিডিটি ভেরি হাই। কেন না? ইন দ্য ডায়াবেটিস there is a hyperglycemia, and that high blood glucose, this high blood glucose can able to bind with the protein molecules of that vascular endothelium. This is the vascular endothelium, and this is the protein molecule, and the high blood level of the glucose can able to bind with it. This is the red mark as the glucose, and that process is the glycation. So vascular endothelium become glycation, and when that glycation is noted in the pulmonary capillaries, so pulmonary capillaries, in case of the diabetic patient, the pulmonary capillaries undergoes glycosylation process, and it becomes narrower. And due to the narrowness, due to the luminal diameter, due to the narrowness of the luminal diameter of the pulmonary blood vessels, the gaseous exchange process will be decreased, and thereby oxygen saturation is also diminished. তার জন্য আমরা বলছি যে আমাদের ক্ষেত্রে একে দেখো কোভিড SARS-CoV-2 ভাইরাস লাংসকে অ্যাটাক করছে, আবার ওদের ক্ষেত্রে অলরেডি ক্যাপিলারি বেডে লুমিনাল ডায়ামিটার কম, তাহলে গ্যাসাস এক্সচেঞ্জ কম হচ্ছে, তাহলে অক্সিজেন স্যাচুরেশন আমাদের আরো কমে যাচ্ছে। এন্ড ফর দিস রিজন যদি ডায়াবেটিক রোগী কোভিড-19 সংক্রমিত হয় তাহলে তার ক্ষেত্রে লাইফ রিস্কটা খুব হাই হয়ে যায়। অ্যানাদার ওয়ান ইজ দ্যাট আমাদের সেলগুলো কি হয় না, ইন কেস অফ দ্য ডায়াবেটিক কন্ডিশন দে আর আনএবল টু ইউটিলাইজ গ্লুকোজ। তাহলে এনার্জি তারা কার থেকে পায় না, বাই দ্য ইউটিলাইজেশন অফ দ্য ফ্যাট। তো ফ্যাট ক্যাটাবলিজম ইজ ইনক্রিজড ইন সেল ইনক্লুডিং দ্য ইমিউন সেল। তাহলে ইমিউন সেলের মধ্যেও আমাদের কি হচ্ছে ডায়াবেটিক কন্ডিশনে, গ্লুকোজ ইউটিলাইজেশন ইজ লো but that fatty acid utilization is high. So that acidic environment in immune cells can able to interfere the performance of the T cell, B cell, the cooperation between the T cell and B cell, both are decreased. তারপরে ইমিউনো অ্যাক্টিভিটিটা আস্তে আস্তে কমে যায় ইন কেস অফ ডায়াবেটিক কন্ডিশন। তারপরে SARS-CoV-2 অ্যাটাক হলে আমাদের সেখানে আমরা প্রোটেকশন নিতে পারছি না। Similarly, neutrophil activity, macrophage activity, CD4 plus activity, all are decreased. So these are the actually the different immune activity, cellular immunity, humoral immunity, both are decreased in the diabetic condition. And thereby the fatality rate, morbidity rate in COVID-19 of diabetic condition, that is the comorbidity very high. So what is our duty? Our duty is number one, that diet pattern should be changed. And we always shift from immuno weakening to immuno boosting diet pattern. We try to avoid the immuno weakening diet pattern. We try to adopt immuno boosting diet pattern. What are the immuno boosting diet pattern? The green vegetables, root vegetables, fruits, green tea, honey, fiber rich food, that should be consumed at regular basis. And we try to avoid fast food, junk food, processed food, preserved food, which are immuno weakening foodstuffs. Similarly, to lifestyle modification, we try to avoid unhealthy lifestyle. What is this? There is a physical inactivity. সবসময় বসে থাকবে এটা ভালো না। Physical activity হয়, তবে আমাদের ইমিউন সিস্টেম অ্যাক্টিভেট হয়। মিনিমাম স্লিপ, ইট ইজ আনহেলদি লাইফস্টাইল। ইরেগুলার স্লিপ, কখনো কোনো রুটিন অনুযায়ী স্লিপ হচ্ছে না, কোনো ইরেগুলার স্লিপ, দ্যাট ইজ আনহেলদি লাইফস্টাইল। অ্যাংজাইটি, দিস ইজ অ্যানাদার আনহেলদি লাইফস্টাইল। ইরেগুলার ফুড ইটিং প্যাটার্ন, খাওয়ার কোনো নির্দিষ্ট সময় নেই, ইজ আনহেলদি লাইফস্টাইল প্যাটার্ন, দ্যাট আর অলসো ইমিউনো উইকিং। তাহলে আমাদের কি করতে হবে না, থ্রু ইনফরমেশন, এডুকেশন এন্ড কমিউনিকেশন উই মাস্ট শিফট ফ্রম আনহেলদি লাইফস্টাইল টু হেলথ ফ্রেন্ডলি লাইফস্টাইল। আমাদের অ্যাটলিস্ট ওয়াকিং দরকার 30 মিনিট পার ডে, অ্যাটলিস্ট কম, স্লিপ 7 to 8 hours পার ডে। কারণ এই স্লিপ হলে আমাদের নিউরো এন্ডোক্রিন ইমিউনো নেক্সাস, নিউরো এন্ডোক্রিন ইমিউনো crosstalk, this is going on, and during this sleep the neuroendocrine immuno crosstalk is responsible for immunoboosting. Timely eating, নির্দিষ্ট সময় খেতে হবে, ওয়াক আপ টাইমলে নির্দিষ্ট সময় ঘুম থেকে উঠতে হবে, স্লিপ টাইমলে নির্দিষ্ট সময় ঘুমাতে হবে, and these are all the biorhythms which are responsible for proper crosstalk among neural system, endocrine system and immune system. And due to the proper operation of this crosstalk, you can able to result immunomodulation properly, positive immunomodulation properly. So I have already mentioned that UV light and UVB light exposure, আমরা বলেছি আমাদের এই যে আমাদের বলছি আমাদের হার্ড ইমিউনিটি, কমিউনিটি ইমিউনিটি, পাবলিক ইমিউনিটি অর সোশ্যাল ইমিউনিটি অর পপুলেশন ইমিউনিটি, এর পজিটিভ পয়েন্টগুলো কি কি আমাদের দেশের ক্ষেত্রে আমরা ইজিলি হার্ড ইমিউনিটি বা পজিটিভ ইমিউনিটি বা পপুলেশন ইমিউনিটি গড়ে তুলতে পারি। এর পিছনে কারণগুলো কি? Now, first positive point from geographical points. Geographical points, that the latitude and longitude, where the UVB light exposure is very high in respect to Europe and American countries. Next one, 90% of the population is below 60 years. আমাদের যদি আমরা ডেমোগ্রাফি দেখি, তাহলে আমাদের ডেমোগ্রাফি স্টাডি করলে আমরা বলছি 90% population is below 60 years. Where COVID fatality rate is less than 3%, is less than 3%. And up to 30 years age group, that fatality is 0.3%. তাহলে দেখো আমাদের ডেমোগ্রাফি আমরা যদি কম্পোজিশন দেখি, সেক্ষেত্রে আমাদের 60 ইয়ার্সের নিচে আমাদের পপুলেশন 90%, এবং তাদের দেখা যাচ্ছে কোভিড ফ্যাটালিটি রেট খুব কম, আর বিলো 30 ইয়ার্সে কোভিড ফ্যাটালিটি আরো কম। তাহলে সেক্ষেত্রে আমরা ইমিউনোমডুলেশন করে আমরা এটাকে অনেকটা ফাইট করতে পারি, কেমন? অ্যান্ড আমাদের আরেকটা পজিটিভ পয়েন্ট যে আমরা সবসময় ফ্রেশ অ্যান্ড গ্রিন ফুড খাই অন্য দেশের ক্ষেত্রে, ইউরোপিয়ান বা আমেরিকান কান্ট্রিগুলোর থেকে। We are not very much habituated with the preserved food, etc., etc. And that is also positive point for immunomodulation. Though there are some negative point in immunomodulation, এখানে আমরা বলেছি যেমন আমাদের একই জয়েন্ট ফ্যামিলিতে আমরা অনেকে থাকি অনেক সময়, তার ফলে সেক্ষেত্রে ইয়াঙ্গার এল্ডার পার্সন রয়েছে, অতএব there is the exposure of the elder person from that younger in this COVID-19. আমাদের স্লাম এরিয়া প্রচুর রয়েছে এবং এই স্লাম এরিয়াতে আমাদের হাইজিন স্যানিটেশন ভালো নয়, সেই জায়গায় দীর্ঘ একটা আমাদের ভয় রয়েছে। আর 22% যেটা সব থেকে আমাদের খারাপ নেগেটিভ পয়েন্ট মানে হার্ড ইমিউনিটির ক্ষেত্রে, 22% অফ দ্য ইয়াঙ্গার সাফারিং ফ্রম হাইপারটেনশন। আমাদের লাইফস্টাইলটা আমরা ভালো ফলো করি না। দেখো 22% অফ দ্য ইয়াঙ্গার সাফারিং ফ্রম হাইপারটেনশন, and then hypertension is a comorbidity for COVID-19. 4% younger suffering from diabetes, and diabetes is another comorbidity of COVID-19. 33% tobacco smoking. 22% of the younger are habituated in tobacco smoking, and that tobacco is also immuno-weakening factor. Okay, so these are the negative points. In spite of that, we try our level best to increase our immunity. And this is the one model for the আমি একটা মডেল দেখাচ্ছি যে হার্ড ইমিউনিটি কিভাবে আমাদের প্রোটেকশন দেয়। এই যে একটা ফার্স্ট স্টেজ অফ দ্য কমিউনিটি, যেখানে আমাদের কি হেলদি ইন্ডিভিজুয়ালস, ইমিউনাইজড ইন্ডিভিজুয়ালস অ্যান্ড ইনফেক্টেড ইন্ডিভিজুয়ালস। ইনফেক্টেড দুজন আছে এই যে রেড গুলো, আর আমাদের হেলদি ইন্ডিভিজুয়ালস ব্ল্যাক গুলো। আমাদের ইমিউনাইজড ইন্ডিভিজুয়াল নেই। এখানে গ্রিন গুলো হচ্ছে সেকেন্ড কন্ডিশন অফ দ্যাট কমিউনিটি। যখনই আমাদের গ্রিন গুলো আসতে শুরু করলো তখন দেখা যাচ্ছে স্প্রেডটা আর হচ্ছে না। আবার যখন গ্রিনটা আরো বেড়ে গেল, মানে ইমিউনাইজ পার্সন আরো বেড়ে গেল, হার্ড ইমিউনিটির দিকে আমরা এগোচ্ছি। আমরা বললাম যে বিভিন্ন ডিজিজে কিন্তু হার্ড ইমিউনিটির কাভারেজ পার্সেন্টেজ আলাদা আলাদা হবে। বিশেষ করে আমরা বলছি কোভিড-19 ক্ষেত্রে আমাদের অ্যাচিভ করতে হবে 78 to 80%। মানে 100 জন ব্যক্তির ক্ষেত্রে 78 থেকে 80% যদি আমাদের অ্যান্টিবডি ডেভেলপ করে যায়, তাহলে আমরা হার্ড ইমিউনিটি পেয়ে যাই। তাহলে স্প্রেডটা আমরা কমাতে পারছি। দ্যাট গ্রিন আর নোন অ্যাজ ইমিউনাইজ পার্সন। তো একটা কমিউনিটিতে যখনই ইমিউনাইজ পার্সন বেড়ে যাচ্ছে তখনই দেখি রেডটা স্প্রেড করছে না। ইজ দ্য মোস্ট ইম্পর্টেন্ট। এবং এখন আমাদের ওয়েস্ট বেঙ্গলে আমাদের কতটা আমরা অ্যাচিভ করেছি 61%। কালকের ডাটা অনুযায়ী 61% আমাদের হার্ড ইমিউনিটি চলে এসেছে। এই যে আমাদের যদি 78%, 80% চলে যায় অক্টোবর নভেম্বরের মধ্যে, তাহলে আমরা কিন্তু এই সুপার স্প্রেডার ডেল্টা স্ট্রেনের এগেইনস্টে অনেকটা ফাইট করতে পারবো। আর নইলে কিন্তু আমাদের অবস্থা খুব খারাপ। যাই হোক, তার জন্য আমাদের এই জন্য দেখছো যে আমরা এখনো লকডাউন থেকে হঠাৎ করে খুলে দিচ্ছি না। লকডাউনটা খুলতে গিয়ে স্টেপ বাই স্টেপ নিচ্ছি। যাই হোক, আমি আর বেশি সময় নেবো না। সো হোয়াট ইজ আওয়ার স্লোগান? আমাদের এই স্লোগানটা কি? আমরা 4টে স্লোগান আমরা বলছি। নট ফিয়ার বাট হোপ। আমরা কোভিডকে ভয় পাবো না। ভয় পেলে আমাদের কিন্তু ওই নিউরো এন্ডোক্রাইন ইমিউনো অ্যাক্সিসটা পুরোটা নষ্ট হয়ে যাবে। সো নট ফিয়ার বাট হোপ, আশা রাখতে হবে। আর দ্য বিগেস্ট ভাইরাস ইজ নট দ্য করোনা ভাইরাস বাট ফিয়ার। নেক্সট স্লোগানটা হচ্ছে দ্য বিগেস্ট ভাইরাস ইজ নট দ্য করোনা ভাইরাস। করোনা ভাইরাস বিগেস্ট ভাইরাস নয়, বিগেস্ট ভাইরাস হচ্ছে ফিয়ার, ভয়। এই ভয়টা করলে আমাদের ইমিউনিটি আস্তে আস্তে ড্রপ ডাউন হয়। ইমিউনিটি যদি উইকেনিং হয়ে যায় তাহলে উই আর ভেরি মাচ সাসেপ্টেবল ফর দ্যাট কোভিড-19। আর নেক্সট হচ্ছে আওয়ার ডিউটি টু স্টপ প্যানিক। প্যানিকটা আমাদের কমাতে হবে। আওয়ার ডিউটি টু স্টপ প্যানিক। So this is our slogan in respect to that lecture. Okay, so I offer my heartiest thanks to all of you, and I do not know that whether my students will be benefited from this lecture or not. If you have any question on this lecture, you may ask. Thank you to all. Okay. Hello.
Thank you, sir. Thank you so much, sir, for such an informative and relevant session in this pandemic situation. Thank you once again, sir.
স্টুডেন্টসদের কিছু কোশ্চেন আছে, বলবো?
হ্যাঁ, বলো বলো।
স্যার, হ্যাঁ, পোস্ট কোভিড হেয়ার ফল কজেস।
পোস্ট কোভিড
হেয়ার ফল কজেস।
হেয়ার ফল
কজেস
বোঝা যাচ্ছে না। হেয়ার ফল বুঝতে পারছি না।
হেয়ার ফল কজেস স্যার। পোস্ট কোভিড হেয়ার ফল কজেস।
কাভারেজ, হেয়ার ফল কাভারেজ।
কজেস স্যার।
কারণটা বলছে বোধহয়, কারণ
হেয়ার ফল কজেস।
Yes, sir. Hair fall causes.
ও
Post
হেয়ার
COVID
ফল
hair.
হ্যাঁ। শোন, হেয়ার ফল কজেসের জন্য তো ডাইরেক্ট করে এখানে যে আমাদের বললাম একটু আগে যে দেয়ার আর সো মেনি প্রো ইনফ্লামেটরি সাইটোকাইনস, এবং তার ফলে বডির মধ্যে যে অ্যাকশন গুলো হচ্ছে প্রো ইনফ্লামেটরি সাইটোকাইন স্টর্ম, এবং এই সাইটোকাইন স্টর্মের ফলে আমাদের হেপাটিক ফাংশন, আমরা বলেছিলাম মাল্টি অর্গান ফেলিওর, হেপাটিক ফাংশন আর অলসো ড্যামেজ, লিভার ফাংশন যেটা আমরা বলছি লিভার ফাংশন ইজ অলসো ড্যামেজ, and that liver function is also most important factor for that তোমার হচ্ছে activity, sustainability, validity of that hair follicle, that is the cell of the hair. So due to the alteration of the hepatic activity, it may result the hair fall, though the proper information, proper research is not known to us till now in this aspect. এটা আমরা এখনো জানতে পারছি না, যদিও একটা সিমটমের মতো দেখা যাচ্ছে, কিন্তু হোয়াট ইজ দ্য রিজন, কিন্তু সাসপেক্ট করা হয় যে হেপাটিক ফেলিওর মে বি ডিউ টু দ্যাট SARS-CoV-2 ভাইরাল ইনফেকশন। হ্যালো
স্যার, আর একটা কোশ্চেন আমাদের ইউটিউব থেকে এসেছে, হুইচ ফুড আর নেসেসারি নাও টু ইনক্রিজ আওয়ার ইমিউনিটি পাওয়ার?
হুইচ ফুড
আর নেসেসারি নাও টু ইনক্রিজ আওয়ার ইমিউনিটি পাওয়ার?
আমরা বললাম যে তোমাদের হচ্ছে গ্রিন লিফি ভেজিটেবলস বেশি করে খাবে, ফ্রুটস খেতে হবে বেশি করে, এবং এই ফ্রুটস গুলোর মধ্যে দেখতে হবে যেন আমাদের হচ্ছে ইমিউনো বুস্টিং নিউট্রিয়েন্ট, যেমন ফার্স্ট ক্লাস প্রোটিন, যেমন আমরা অ্যানিমাল প্রোটিন খাবো, ফার্স্ট ক্লাস প্রোটিনের মধ্যে কোনটা আমাদের ইমিউনো বুস্টিং করে না? ফার্স্ট হচ্ছে এগ প্রোটিন, এগ হোয়াইট প্রোটিন, তারপরে মিল্ক প্রোটিন, ফিশ প্রোটিন অতটা করে না, মিট তো নয়, তো ফার্স্ট হচ্ছে এগ হোয়াইট প্রোটিন and then the milk protein. Similarly, there is the leafy vegetables, root vegetables, fruits. These are most important, and vitamins, vitamin C, vitamin D, zinc, glutathione. These are the most important positive immunomodulators. Positive immunomodulators which are responsible for positive immunomodulation. এছাড়াও আমি বলেছি ফুড স্টাইলের ক্ষেত্রে যে প্রপার টাইমিং এ খেতে হবে। বায়োরিদম, কোনো বায়োলজি, কোনো বায়োলজি মানে আমাদের বডির মধ্যে একটা বায়োলজিক্যাল ক্লক আছে, রিদিম অনুযায়ী প্রত্যেকটা ঘটনা যদি আমরা করতে পারি ঠিক মতো, তাহলে কি হয় না, we can able to control that and coordinate the neural system, endocrine system and immune system, and as a result the positive immunomodulation can be executed. কেমন? টাইমলি খাওয়া, টাইমলি ঘুমানো, ফাস্ট ফুড বাদ দেওয়া, ফ্রেশ ভেজিটেবলস খাওয়া, ফ্রেশ ফুড খাওয়া, এগুলো হচ্ছে আমাদের। তার মধ্যে আমরা কি কি বললাম? ফার্স্ট ক্লাস প্রোটিনের মধ্যে আমরা বললাম কোনগুলো, তারপরে বলা হচ্ছে ফ্রুটস, তারপরে বললাম লিফি ভেজিটেবলস, রুট ভেজিটেবলস, নেক্সট হচ্ছে that the vegetables containing high level of zinc, high level of vitamin C, high level of vitamin D, high level of vitamin E, vitamin A. These are all the immunoboosting. ঠিক আছে?
ওকে স্যার, থ্যাংক ইউ। থ্যাংক ইউ।
Thank you, sir.
We are going to another session. Now I would like to request Shati Bhuya Madam, faculty member of Nutrition Department and joint convener of this webinar, to welcome Dr. Shukanto Mondol sir. Over to you, ma'am.
Thank you, Amit sir. Welcome to all, my respected principal madam, respected resource person, my colleagues and beloved students. We are very grateful to have with us Dr. Shukanto Mondol sir. He is currently working as Principal Scientist of Animal Physiology Division at ICAR, National Institute of Animal Nutrition and Physiology, Bangalore. He has completed his PhD on molecular characterization of prostaglandin synthase genes in buffalo from ICAR IVRI in 2006 and did postdoctoral assignment from University of Laval, Canada in 2010. He has been working in the field of genomics of hormones, reproduction and stress for last 24 years. He also involves in many other researchers in various disciplines. Sir has published 132 research publications, written 13 book chapters, and edited 3 books published by Elsevier. Sir has received many prestigious awards like Overseas Associateship Award, long term by Department of Biotechnology, Government of India, Professor G.P. Talwar Mid Career Scientist Award, Professor G.K. Pal Award, Sir Siri Research Award, Dr. K. N.G. Reddy Award, Professor P.B. Sen Memorial Oration Award, and ISSRF Young Scientist Award, etc. He is honored with the Fellow of National Academy of Veterinary Sciences, Fellow of Indian Association of Biomedical Scientists, Fellow of Society for Applied Biotechnology, and Fellow of Indian Council Society. He is editor-in-chief of current research in poultry science and also editor of many other national and international journals. I would like to request you, sir, to give your valuable speech. Over to you, sir.
Good afternoon, ma'am. So, respected chairperson, is it audible?
Yes, sir. Audible,
Yes,
sir.
sir.
Acha, Then, respected chairperson, distinguished expert and resource person, and all the organizers and the students. Today, I am here to deliver my talk on reproductive and nutritional strategies for early embryonic loss. At the outset, I would like to express my sincere gratitude to Dr. Rupa Dasgupta, a principal of the college, for giving me an opportunity to talk before the students. And my special thanks also Professor Dr. Devidas Ghosh and Snigdha Sahu for coordinating the events. Now I am presenting my speech on the reproductive and nutritional strategies for minimizing early embryonic losses. In a report on the cattleside.com, Professor Henrik Bolwin from University of Veterinary Medicine, Germany, he reported that declining fertility is a problem in the food-producing animal. Early embryonic mortality is one of the major reproductive problems for the domestic mammals, including humans also. Now, why this embryonic losses is a major concern? Because of huge production failure in case of large ruminants, specifically cattle, buffalo, and also small ruminants like sheep and goat. And this majority of the losses occur because of the failure of the dialogue between the mother and the conceptors. Now, before coming to the subject proper, we should know that the embryonic mortality is categorized into three categories. One is the early embryonic mortality, which occurs before day 28 of the pregnancy, and it doesn't affect the length of the estrous cycle. Even the signs of the abortions are not diagnosed. Second category is late embryonic mortality, which occurs between day 28 to 42 of the pregnancy. And it is diagnosable because in some cases the embryo and membranes are aborted, and the obvious sign is the animal will come to the estrus. And the third category is the fetal loss, which occurs after day 42 of the pregnancy. Now these are how the embryos, initially there will be fertilization. After fertilization, then from 1 cell stage, then 2 cell, then 4 cell, then 8 to 16 cell, then 32, and the morula and the blastocyst stage. I think all of you know these events, I think. Then coming to the early embryonic development, generally 2 cell is developed on day 1 and 4 cell is developed around day 1.5 after fertilization, and it reaches blastocyst around days 7 to days 12, and then followed by the elongated blastocyst around days 13 to 35. And by day 30, we have observed that there are 35% losses. So this 35% is a huge loss, particularly the livestock production. Now, if we show the losses, generally these are the different days of the pregnancy, day 0 to day 22 of the pregnancy. And we have seen, it has been reported by many authors like Thatcher, Peter, and Humboldt. They are the stalwart in this field, and they have found there is 10% losses on day 0, that is the day of estrus and fertilization. Then after day 5, the embryo enters into the uterus at the stage of morula. After that, the embryo reaches the blastocyst stage, and then followed by the blastocyst hatching on day 8 of the pregnancy, followed by maternal recognition of pregnancy between day 13 to day 16. Now, what is the maternal recognition of the pregnancy? Maternal recognition, that means mother recognizes that she is pregnant. So the thing is that there is some signaling molecule is coming from the fetus, and that is giving some biochemical signal. Because of that, we can easily identify that the animal is in and the maternal recognition of the pregnancy. Then the embryo attached to the endometrium by around day 20 to day 22. Now, if we see the cumulative losses, generally there are 10% losses on day 0, that is the day of estrus. 30% losses has been found between day 2 to day 7 of the pregnancy, and most of the losses, that is 40% losses, has been observed between day 8 to day 17 of the pregnancy. Now, naturally, question come, why early embryonic loss is a concern? Because of a loss of valuable offspring and their production, increased maintenance cost of the animal, because we have to maintain the animal. It hampers the genetic progress and it hampers the advances in assisted reproductive technology. Now, if we see the causes, there are 8 causes that are responsible for the early embryonic mortality. The first one is genetic abnormalities, then nutritional stress, then the uterine environment, inappropriate insemination, reproductive management program, endocrine imbalance, thermal stress. So there are mainly 8 factors that are causing the early embryonic losses. So I'll come one by one the causes. The first is the genetics. Mainly 5 to 10% losses occur because of the genetic abnormalities, and that too during the first 2 weeks of the pregnancy. So naturally, what happens if there is abnormal chromosomal number? So there will be the abnormal growth of the embryo, and death will be there during the first trimester of the gestation. What happens? It has been reported by Gary that there are 3 to 5% losses occurs in bovines due to chromosomal defect. Now, JV, he reported that if there is an autosomal recessive gene for uridine 5' monophosphate synthase, if it is present in the homozygous condition, it causes the losses of the embryo before day 40 of the pregnancy. Now, coming to the heat stress, that is the second most important cause for the embryonic losses. Mainly 10% losses occur because of heat stress before day 25 of the pregnancy. Roth et al., they have seen that if animals are exposed to heat stress during early follicular stages of development, so it affects the oocyte quality and subsequent embryo development will be affected. Even in vivo heat stress between day 8 and day 16 of the pregnancy, it compromises the luteal function in terms of there will be increased production of prostaglandin F metabolite, that is PGF2 alpha, and because of that corpus luteum cannot be maintained during pregnancy, and because of that there will be abortion or embryonic losses will be there. Now this diagram explains what happens during heat stress. So when the animals are exposed to heat stress, so naturally they will consume the less food, so there will be reduced dry matter intake, and because of that negative energy balance will be there. Due to negative energy balance, decreased GnRH and LH secretion from pituitary and hypothalamus. Because of that, there will be low estradiol production from the ovarian source, which ultimately leads to poor estrus expression and poor quality of the oocyte, leading to infertility. Simultaneously, when there is heat stress, the animal uterine environment itself gets compromised in terms of the losses, embryonic losses or abortion. So both these things will contribute to the infertility. Now coming to the heat stress, what is heat stress? Heat stress is that particular point when the animal cannot dissipate adequate quantity of the heat to maintain their body thermal balance or the normal body temperature. In addition to the heat stress, when there is heat stress, so in association with the heat stress, there will be oxidative stress also in association with the metabolic stress. So when there is oxidative stress, there will be more production of the reactive oxygen species, particularly the hydroxyl atoms, and they are not bad. We have a very bad idea of that the reactive oxygen species, they are very bad, but they are not bad. They are required for some biological functions like sperm DNA condensation and ovulation too. But when there is heat stress, this reactive oxygen species concentration reached the threshold level. So what would happen? Because of the threshold reactive oxygen level, there will be lipid, protein and DNA damage, which ultimately leads to the cell injury, and it inhibits the embryonic development. So there will be the degeneration of the embryo. So because of heat stress, there will be the embryonic losses. Now coming to the third important factor that is responsible for embryonic losses is the nutritional stress. Generally, if animals they are gaining the body condition during pregnancy, they will have the less embryo mortality. But when the animals they lose their body condition during pregnancy, so there will be probability of higher losses. What happens? This nutrition mainly affects the ovulation rate and sexual activity in case of sheep. Even we have seen when the animals are exposed to combined heat and nutritional stress, so they will take less food, less water, so respiration rate and rectal temperature are also significantly affected. And this causes significant losses because that I will be showing in my later slide. And one more problem, Butler, he reported that if we feed too much protein diet, what happens? There will be high concentration of ammonia and urea if we feed high protein diet. That will interfere with the normal action of the progesterone. So they cannot maintain the uterine environment. So naturally the embryo or the abortion will take place during early pregnancy. Now the reproductive management, that is the timing of the insemination, is another important factor. If we inseminate too early, what will happen? The embryo quality will be high, but the fertilization rate will be low. On the contrary, if we inseminate at later stage, very later stage, what will happen? The embryo quality will be low, but the fertilization rate will be very high. So we should have a optimum timings of the insemination, and that optimum timing is 6 to 18 hours after onset of estrus. So when the animal is showing heat symptom, particularly large animals, particularly buffalo, they will show in the early morning or late night. So no farmers can detect whether the animal is in heat stress, whether the animal is in heat or not. So what we have to do in the next morning, in between 6 to 8 hours after onset of estrus, you have to inseminate the animal. Now coming to the hormonal imbalance, that is another important cause. I think all of you must heard about the progesterone. This progesterone is an important hormone for maintaining the pregnancy. Now there are two major reasons why the progesterone is lacking. The first important condition is that corpus luteum will be there, but it will be having the short lifespan. So what will happen? So lipolysis, corpus luteum will regress before embryo sends the signal that embryo is present. So naturally the embryonic losses will be there. And the second condition is that corpus luteum will be there and that will be having the normal lifespan, but they will secrete low progesterone. But we need high progesterone for maintaining the pregnancy, and because of the low progesterone, the pregnancy cannot be maintained. Even we have seen in buffaloes, particularly I have seen during my master's program, we have seen if there is inadequate luteal function in buffalo, so there will be the more chances of embryonic losses. So what is inference from this one? We should have sufficient quantity of the progesterone during early pregnancy. So Robinson et al., they supplemented progesterone to the cows, and they found there are less chances and the embryo survivability was more. Now coming to the infectious factors, that is the particularly Corynebacterium pyogenes, they cause abortion and endometritis in cattle. Vibrio cause infertility and embryo mortality. Brucella abortus is another important factor which mainly affects during mid to late pregnancy and causes abortion. And lastly, the bovine herpes virus, it causes the embryonic losses and pathological changes in the uterus during pregnancy. Now, asynchrony between the conceptus and the maternal system is another important cause. Why the embryonic losses is taking place? Because of the failure of the dialogue between the mother and the conceptus. The signal that conceptus is sending, maybe it is not reaching to the mother. So because of the failure of the signaling, and that period is called maternal recognition of pregnancy. Generally, maternal recognition of pregnancy in case of cow, days 16 to days 19 of the pregnancy. In case of you, it is 2 to 3 days less, that is day 12 to day 13 of the pregnancy. And in goat, it is around day 7 of the pregnancy. Now, this diagram shows the relationship between embryo, uterus, ovary during maternal recognition of pregnancy. Now we know, I think you must know the ovary, the corpus luteum is present in the ovary. So this corpus luteum progesterone produces progesterone, and this progesterone maintains the pregnancy. This I think all of you know. Now what happens? Mainly endometrium produce prostaglandin F2 alpha from arachidonic acid. So what this prostaglandin F2 alpha does? This prostaglandin F2 alpha cause regression of corpus luteum, and because of that embryo cannot stay inside the mother uterus, ultimately leading to the abortion. So our target should be to stop the production of prostaglandin F2 alpha. Now what happens during pregnancy? The corpus luteum will be very big size, so that corpus luteum produce progesterone, and that progesterone prevents the synthesis of prostaglandin F2 alpha during pregnancy by inhibiting the synthesis of oxytocin receptor. I think you must have heard about the oxytocin, estrogen, and progesterone receptors. So they are having great role during maternal recognition of pregnancy. What happens during pregnancy? The progesterone that is produced from the corpus luteum, it prevents the synthesis of oxytocin and estrogen receptor. So estrogen and oxytocin cannot bind to the progesterone, to their receptor, and they cannot produce the prostaglandin F2 alpha. In that way, the prostaglandin F2 alpha production is less, so the pregnancy is maintained. Now, if we see the embryo's contribution, what happens? The embryo produces some signal that is called interferon tau between day 8 to day 17 of the pregnancy. So that interferon tau prevents the synthesis of oxytocin receptor and stops the production of PGF2 alpha. Thereby the pregnancy is maintained. Now I think you must have heard about the prostaglandin. So what that prostaglandin does? This prostaglandin was identified and got the Nobel Prize by von Euler in 1934. This prostaglandin plays an enormous role in reproduction, various reproductive hormones like estrocycle, maternal recognition of pregnancy, implantation, and parturition. So these are the hormonal events that are taking place during estrocycle. What is happening when there is after ovulation? I think you must have heard about the ovulation. Ovulation means when there is shedding of ova from the ovary. So after ovulation, corpus luteum start developing, and with the development of corpus luteum, the progesterone production will be high. And around day 6 to day 7, the progesterone reaches its plateau, and then that plateau maintained up to day 15 or day 16 of the cycle. And then because of the PGF2 alpha pulses around day 14 to day 16, the regression of the corpus luteum will be there and the progesterone production will be low if the animal is in cycle, estrous cycle, or if the human is in menstrual cycle. These are the events happening. Now what is happening when the animal is pregnant? So the progesterone concentration should be high, and this is high because around day 14 to day 18, the embryo is producing signal that is called interferon tau, and this interferon tau is preventing to produce PGF2 alpha. And this PGF, since the PGF2 alpha is less, so they cannot regress the corpus luteum, and thereby the corpus luteum structure is maintained, and they produce the more progesterone during pregnancy. Like that, the pregnancy is maintaining because the embryo is giving some signal, and that signal they are giving during maternal recognition of pregnancy. Now, this diagram shows the mechanism happening during establishment and maintenance of pregnancy. Generally, progesterone, estrogen, and oxytocin receptor will be there in the uterus. Now, what is happening during establishment of pregnancy? So that embryo produce some signal. In particularly in bovine, it is called trophoblastic protein. The trophoblast of the embryo produce some protein, which later identifies as an interferon tau. So these trophoblastic protein, they will bind to the receptor which is present on the uterus, and they stabilize the progesterone receptor. They stabilize the progesterone receptor and decrease estrogen receptor and oxytocin receptor. So because of decreased synthesis, so there will be less production of PGF2 alpha, thereby the pregnancy is maintained. Pregnancy is established and the pregnancy is maintained because of both the events are taking place. Now coming to the prostaglandin, I spoke to 2 slides before that prostaglandin they are having enormous role in pregnancy. Now this diagram shows the production of the different prostaglandin because the different prostaglandin are having different role during pregnancy. Now most of the prostaglandins are synthesized from arachidonic acids, then they get converted into PGH2 under the influence of COX2. Then PGH2 under the influence of prostaglandin F synthase gets converted to PGF2 alpha. And this PGF2 alpha is a culprit molecule. So during pregnancy, this PGF2 alpha should be low. So I will show in my later slide that we have taken a different approach. We have edited or knock out this PGF2 alpha during pregnancy so that the pregnancy can be maintained. That I will show in my later slide when we will explain the targeted genome editing events for the prostaglandin F synthase gene. Now PGH2 gets converted into PGE2 under the influence of prostaglandin E synthase, and this PGE2 is beneficial. This because this PGE2 it maintains the structural integrity of corpus luteum, so the corpus luteum will be maintained during pregnancy. They will produce more PGE2. They will produce more progesterone, so the pregnancy will be maintained. So this PGE2 is very, very beneficial biomolecule for our research. Now I would like to share some of our research. We studied the effect of heat and nutritional stress during maternal recognition of pregnancy. What is happening in the uterus? Very quickly I will explain. So this is the stress model we have developed in the climatic chamber. Inside the climatic chamber, the temperature was maintained by manipulating the temperature, and that temperature we adjusted as per the outside climate chamber. Suppose outside climate chamber was around 38, 10 o'clock, so we adjusted the temperature inside the climate chamber 38 degree at 10 o'clock, 40 degree at 11 o'clock, 42 degree at 12 o'clock, at 1 o'clock 43, at 2 o'clock 44 degrees centigrade. Then the temperature was reduced at 3 o'clock to 42 degree because outside temperature was 42 degrees. So we simulated the outside temperature inside the climate chamber, and this was for 6 hours. The animals were exposed to this temperature, and we found there was decreased ovulation rate, the ovarian response, superovulation rate, different biochemical parameters like glucose, cholesterol, and protein. Hormones like T3, T4, and estradiol levels also low. Even the duration of the estrus, the animal that we were showing, was low. But there was the concentration of hemoglobin, packed cell volume, and hormone like cortisol and progesterone was higher. Then we have seen the morning and afternoon, the respiration rate, pulse rate, and rectal temperature. And we found that in heat stress animal, there was significant increase in respiration, rectal rate, and rectal temperature, and the pulse rate. Even the plasma cortisol level, progesterone level was higher in the stress group as compared to the control group. Then we studied some of the genes that are mainly important for maintaining the maternal recognition of pregnancy, because that is the crucial event. And because of that only embryonic losses are happening. So what we have done, we have artificially induced cycle. We have artificially induced by inserting progesterone sponge inside the vagina to the animal, and that progesterone sponge was kept inside for 12 days. On day 12, we took out the progesterone sponge, and then we injected 200 IU pregnant mare serum gonadotropin. And after that, hand mating was done on day 13 to day 14. And then the animals were maintained as a pregnant animal, and then they were exposed to heat stress, as I mentioned in my earlier slide. And then what we did, the animals were slaughtered on day 11, day 13, and day 50 of the pregnancy. Actually, we have not slaughtered. We have by laparotomy, we have opened that one, and we have taken out the tissue by using the laparoscopy, because we want to study some of the genes, and these are mainly maternal recognition of pregnancy are happening between day 12 to day 14 of the pregnancy. Now we have seen very interestingly, prostaglandin E synthase is an important gene which produces prostaglandin E2. So naturally it should be high during pregnancy. But what is happening? When the animals are exposed to heat stress, you see on day 11, day 13, and day 15, in heat stress group it decreased. That shows that heat stress is causing the abortion. So the animal should not be under heat stress during pregnancy. Even we have seen the contrary report on prostaglandin F synthase gene. It is high on day 13 and day 15. Why it is high? And it must be high because prostaglandin F synthase should be high, because if prostaglandin F synthase expression is more, there will be more prostaglandin F2 alpha production, and that prostaglandin F2 alpha will cause abortion or embryonic losses. So that's why it is high in heat stress group. And we have also seen in the heat stress group also the embryonic losses was more. Similarly, we studied the COX2 and osteopontin gene. These are also maintaining for implantation, and we found there was a significant decrease in COX2 gene in the heat stress group in all the 3 days, day 11, day 13, and day 15 of the pregnancy. Osteopontin, there was non-significant decrease in all the days. But galectin and integrin gene, we found there was it was significantly higher during the heat stress group. So these are the important genes that are mainly regulating the maternal recognition of pregnancy events. Then we have studied the nutritional stress effect on ovulation rate, embryo production. We have taken 8 animals in the control, and in the nutritional stress group, we gave 30% less food, maintenance diet. And we found there was mean ovulation rate was slightly higher in the nutritional group, but it was not significant. Supraovulatory response was more, but the embryo recovery was very, very less. It was significantly less in the nutritional stress group. We have also studied the COX2, PGFS, heat shock protein, INOS, SERPIN, PGES, osteopontin, and MX gene. And we found that COX2, PGFS, HSP, INOS, and SERPIN gene expression increased following nutritional stress, whereas PGES, osteopontin, and MX protein is less. It is as per our desire, because PGE synthase expression should be significantly low during nutritional stress. So when the animal is in under nutritional stress, or when the human during pregnancy is under malnutrition, so there will be less production of PGE2, which is required for maintenance of this pregnancy. So because of less prostaglandin E production, the embryonic losses or abortion is taking place in human or animal. In addition to that, we have studied the combined heat and nutritional stress effect in sheep, and we took to 8 animals in the control group and 8 in the combined stress group, and we found there was significant decrease in embryo recovery in the combined stress group. It is as it is expected in combined stress, in the combined heat and nutritional stress, there was significant decrease in the embryo recovery in sheep. And we studied some of the gene expression like COX2, PGES, integrin, galectin, heat supporting, and PGES. Naturally, PGES expression is decreased. It should be low when the animal is in combined heat stress, and because of combined heat stress, the embryo mortality will be very high or abortion will be very high. Similarly, heat shock protein expression is more, and PGFS as desired. So these are mainly causing the embryonic losses. In addition to in vivo experiment, we have studied some of the experiment in vitro. We have seen what is the in vitro heat stress effect on oocyte maturation in sheep. So we have taken a control group where they are exposed to 38, and the heat stress group where the animals were initially exposed to 38 for 6 hours. Then the temperature was increased to around 41, and the other group was around 43 degrees. They were exposed for 18 hours, and we have seen the maturation rate was significantly low, both the heat stress group. It was 37% as compared to the control group, 72%. And in heat stress group 2, that is 43-degree group, it was very significantly low at 1% level. 13.5% was the maturation rate. So in vitro heat stress also significantly affect the maturation rate. So these are the very excellent quality of the oocyte. Excellent means which are having 3 to 5 more than 4 to 6 layer of cumulus layer. Grade 2 is the good quality of the oocyte, which is having 2 to 3 layers of cumulus cell, and fair quality, which is having maybe 1 to 2 layers. And now what is happening after heat stress? You are seeing it became black. the ooplasm is dark, and the shape become from oval, it becomes a different type of shape. Even sometimes the cumulus layer that is surrounding the oocyte is coming out, it is dispersed after the heat stress. Here it is fully degenerated oocytes, and the cumulus cell it is entirely black. So the oocyte, when they are exposed to heat stress, it become black. And next, the shrunken ooplasm. The ooplasm is shrinking. Now we have studied some of the biochemical parameters which are mainly important for the oocyte growth and maturation. And these biochemical parameters are protein, glucose, calcium, phosphorus, chloride, and urea. And we have seen following heat stress, there was decrease in those parameters, which is indicating that it is really decreasing the growth and maturation of the oocyte. Similarly, we studied the heat stress effect on the buffalo endometrial cell. We exposed to different 40 degrees centigrade for 18 hours, and we have found the gene expression like similarly. So I'm not explaining in detail. Now we are coming to the impact of the heat stress on the buffalo oocytes and embryos. We have studied the in vitro effect of the heat stress on the buffalo oocytes and embryos. And this is our stress model, because the oocytes were exposed 40 degrees centigrade, one group 40 degree, other group 42 degree, during different hours of maturation in vitro, 0 to 4 hours, 8 to 12 hours of in vitro maturation, or 16 to 20 hours of maturation, and even after fertilization, 0 to 4 hours after fertilization, then day 5 to day 6 of maturation, and then day 6 to day 7 of fertilization. And what we have seen, the trophoectoderm cell, it gets compromised. In control group, you are seeing the red one is showing that the inner cumulus cell is decreased when the animals are exposed to the heat stress of 40 degree. It significantly decreased. Even in group 2, which are exposed to 40 degree, even for group 2, which are exposed to 42 degrees centigrade. Then we studied some of the gene heat stress gene like HSP8, C2, and MN's superoxide dismutase, and these gene expression were higher following heat stress in vitro. Similarly, maternal to embryo transition gene also we studied, and we found there was significant decrease at 4 and 8 to 16 cell stage. There was significant decrease of PFP2 gene, U2AF gene, and E1F1 gene. These genes are mainly responsible for maternal to embryonic transition gene. Also, some of the genes like jonaploidin, ETS1, and sodium potassium, there was significant decrease in the blastocyst stage as compared to the morula stage following heat stress. Similarly, maternal recognition of pregnancy-related gene also significantly decreased, like TED1, CDS2, GATA3, and interferon tau. They significantly decreased in 8 to 16 cell stage, morula and blastocyst stage. Now, coming to the most important aspects of this presentation is that the CRISPR-based, I think all of you must heard about the CRISPR-based technology, which has come very recently. And because of this technological advancement, last year Nobel Prize was awarded to Professor E. Carpentier. Actually, she is working in Max Planck Institute of Science of Pathogen in Germany. And Dr. Dodna, she is working in University of California, USA. They both were awarded Nobel Prize in Chemistry because they developed a method for the gene editing. So we are using the same methodology in livestock, particularly buffaloes. Before coming to our results, first we should know what is the genome editing by CRISPR-Cas9. What is CRISPR-Cas9? Generally, what happens, there will be a complex that is called single guide RNA, which remains associated with the Cas9 protein. So these particularly complex will travel through the DNA, and wherever they will find a suitable match, they will bind with that, and this Cas9 enzyme will cut the DNA. So there will be a conformational change, and that will activate this Cas9. So what will happen? They will cut, so the double-stranded DNA will be clipped, and this DNA itself will be repaired by the cell. So these are the events that are taking place during CRISPR-Cas9 mediated genome editing events. This is the mechanism of CRISPR-based gene editing system. So these are the DNA base to be changed, what we are targeting. What will happen, that complex guide RNA will remain associated with the complex, it remains associated with Cas9 as a complex. So this complex will bind to that particular, it will travel through the DNA till afterward. And then that will bind to that one and it will cleave. It will cleave that particular, that portion. It will cleave that portion and then there will be a damage will be there. And because of that damage, then what will happen, our DNA template with the desired sequence will bind to that place and they will bind wherever they find the matches that the sequence we are sending that place. So the DNA edited to the desired sequence. So because of this technology is having a lot of application, so there will be the CRISPR-Cas9 mediated editing events, and both color has been taken as a mosaicism because the tyrosine gene was edited, and you are seeing that the black color of the mouse. Even for the liver disease also this technology is being used for the for human purposes, for human beings, for the treatment of the cirrhosis or hepatitis. Even for the organ transplantation, particularly in taken up a very good project from the National Fund that is being funded by our parent organization, IQAC, and where we have targeted 3 different genes like cycloxygene COX-2, prostaglandin E synthase, and F synthase gene, and we have designed 3 different single guide RNA so that we can target those genes. And that single guide RNA we have ligated into this vector, that is the CRISPR expression vector, and then that vector was transformed into the competent E. coli cell. After that, the transformed cells were selected by ampicillin resistant, and then we picked up the positive colonies, and those colonies were further grown for isolation of the plasmid, and then these positive clones were confirmed by PCR using U6 primer, and then we had sent those positive clones to the company fellow for sequencing, so that we want to check whatever the SGN we have inserted, it really inserted or not. So these are the COX-2 single guide RNA. We found these are the patches, and then we isolated plasmid, and then we 10 clones, 10 clones, positive clones we have taken out, and we have done the PCR before sending for sequencing for the confirmation. Then the sequence was sent to the company fellow for the sequencing, and then after sequencing we have seen that those particular sequence are present in whatever we have done in our positive clone. So it shows that correctly our single guide RNA molecule, that single guide RNA molecule will create a change in our desired gene. So that is present, that yellow color shows the found that all clones are positive. Then we sent for sequencing, and we found that our desired single guide RNA was there. Then we studied the effect of this CRISPR-based editing of COX-2 gene, and we found there was significant decrease in PGF2 alpha and PGE2 concentration and the expression of COX-2 gene. So this work was carried out by one of our students. So he has recently submitted thesis, Dr. Dipankar Paul. He has recently worked on this aspect, and he has submitted the thesis, and this thesis is under consideration, and he has done a very good and excellent work, which he will be coming out in terms of very good publication. Simultaneously, we have also targeted the PTGES gene and PTGFS gene, and we found there was significant decrease in PGE2 production after CRISPR-based editing, and there was significant decrease in expression of PTGES mRNA in buffalo endometrial cells. Similarly, PTGFS also, the mRNA expression level we found there was significant decrease, and the PGF2 alpha level also there was significant decrease. Now, currently, one of our cooperating center, I think you must have heard about NDRI Kernel, they are working on the knockout mice. So they are trying to develop. So what they have done the superovulation. Then they have taken out this embryo. They have taken out 2 cell stage embryo at 2.5 days post-coitus. Even morula stage embryos also they have isolated at day 3.5 post-coitus. blastocyst at 4.5. So these are the embryos they have taken out, and these embryo then they have transferred into the surrogate rat, which was extra synchronized, and they transferred by inserting a speculum that they are inserting, and they have come out with the production of embryos. So these are the particularly pups they have produced. This P indicates the pups. They have produced 3 pups, and M indicates the mother. I will show the videos. So these are the. Is it audible? Hello.
Yes, sir.
Yes,
Yes,
sir.
sir.
So these are the pups. It has been produced by So now these pups, when these are the young pups, and when these pups are 11 days old, so these are the pups we produce first time in our country by non-surgical embryo means. Then coming to the strategies, how to minimize these embryonic losses. So first important strategy is development of insemination program. We should do at correct time, so the animal will be exactly in the time of heat, so that the thing will be there. Second is nutritional strategies. Thirdly, injection of the progesterone hormone so that the pregnancy can be maintained, and exogenous administration of luteotropic agent, and lastly the use of bovine somatropin. The first important synchronization protocol is Ovsinch protocol. So this is nothing but injection of PGF12 and GnRH to synchronize the ovulation in animal. Generally GnRH and PG prostaglandin F12 was injected at 7 days apart, and then the animals were inseminated. Using this protocol, we found that the embryonic losses was less. Now, in addition to that, a pre-cinch protocol, which is slightly modification of Ovsinch protocol, where we have given 2 injections 14 days apart, and they are administered 12 days before initiation of the first GnRH injection. So, where we have given 2 doses of prostaglandin injection 12 days apart before the GnRH injection, and we did the time artificial insemination, and we found that embryonic losses was less in this case. Then heat cinch is also another alternative to this pre-cinch protocol, where 1 milligram estradiol cypionate is administered 24 hours after PGF12 injection of Ovsinch. So this is the heat cinch protocol. 2 doses of prostaglandin F12 should be given at 14 days apart, then after 14 days GnRH, and then after 7 days again PGF12, then after 1 day estradiol cypionate should be injected. So these are the different insemination protocol. These are the different extra synchronization protocol. If you follow this protocol, the embryonic losses will be low. Now coming to the nutritional strategies. What has been observed that if we feed too much protein, the number of service is required per conception is more. If we feed 19.3% protein, the number of service per conception will be 2.47. But if we feed the 12.7%, this one, the conversion will be, the service per conception will be very low. So we should feed optimum quantity of the protein. It's not like that we should feed huge quantity of the protein. If we feed huge quantity of the protein, so there will be more production of ammonia and urea, and that will create problems, so that pregnancy will not be maintained. Armstrong et al., they supplied 0.8 kg fish meal, and they found there was improvement in the conception rate in dairy cattle. Now, Robinson et al., they found that if we inject progesterone hormone, so there is the pregnancy rate is improved as compared to the control animal. Even if we inject the luteotropic agent like human chorionic gonadotropin or gonadotropin releasing hormone, on the day of insemination, generally pregnancy rate is improved. Even administration of interferon, because this interferon maintains the pregnancy in different species, it has been observed that in cattle, sheep, if we administer oxytocin, the pregnancy rate is improved. I'm not going in detail. Lastly, the use of bovine somatotropin. This in vitro, it has in this also in vivo, it has been observed in cows that administration of bovine somatotropin, particularly they injected 500 mg Posilac, and they found there was increased in the pregnancy rate as compared to the control animal. In control animal, the pregnancy rate was 42%, but in treated group it was 57%, and which was significantly higher. Even in vitro also it has been observed that the bovine somatropin and IGF-1 it stimulated the in vitro development of the embryo. Now, from our studies, we can conclude that under this project, we have developed the heat stress model by simulating the outside atmosphere inside the climatic chamber. We have developed the nutritional stress, so the animals were fed 30% less as compared to their maintenance diet. And we have seen the heat stress, nutritional stress, and the combined heat and nutritional stress modulated the expression profiling of the genes that are mainly important for MRP and implantation in case of SIP. Even heat stress in vitro affected the growth maturation of the oocyte by altering the ionic and the metabolic content of the oocyte. Exposure of heat stress also in buffaloes has been affected the development of the embryo in case of buffalo. And finally, the targeted editing of COX-2, PTGES, and PTGFS genes significantly decreased their expression in buffalo endometrial epithelial cells. So lastly, I would like to acknowledge our National Fund for giving the funding, Dr. P.K. Agarwal sir, who was the former director of the National Fund. Thirdly, I would like to thank Dr. Rupa Dasgupta ma'am, Dr. Devidas Ghosh sir, and Mrs. Snigdha Sahu and other organizers for giving me an opportunity to talk before this gathering. Next, our director, NDRI, and the director of the NDRI kernel, and our partner institute director, and Dr. Mohan from International Center for Genetic Engineering and Biotechnology for helping our genome editing work, and our cooperating center principal investigator like Dr. D. Malaka, Dr. D.N. Das, Dr. Datta, Dr. D. Kumar, and the students like Dipankar, Praveen, and our SRFs. Thank you.
Thank you so much, sir, for such an informative session. Once again, thank you, sir. Our technical session end here. Once again, we are thankful to our principal madam and our resource persons, Professor Devidas Ghosh sir and Dr. Sukanta Mondol sir. Now, I would like to request Sheikh Sarfaraz Ali sir, Assistant Professor, HOD, Department of Chemistry, and
Amit question answer session sir. Okay.
So there is
Hello?
Yeah.
any question from YouTube link?
Sir, only one question.
Yeah, tell me.
Sir, what causes pain during PCOD?
What causes pain?
Pain.
Because when there is PCOD, particularly in human, so what happens, there will be alteration in the hormonal milieu. So because of that hormonal milieu, and maybe the pain will be there because of prostaglandin. Out of this prostaglandin E2 and prostaglandin F2 alpha, one prostaglandin is causing pain, particularly muscular pain. So maybe because of the hormonal imbalance. Because I have not done research, so I have just guessed that one.
Thank you, sir. Now, our technical session end here. Once again, we are thankful to our principal madam and our resource person, Professor Devidas Ghosh sir and Dr. Sukanta Mondol sir. Now, I would like to request Sheikh Sarfaraz Ali sir, Assistant Professor, HOD, Department of Chemistry, and also Organizing Secretary of this webinar. Over to you, sir.
Thank you, Amit. Am I audible, Amit?
Yes, sir. Audible.
Thank you once again. Good afternoon, all. I deem it great honor and privilege to propose the vote of thanks on this memorable occasion. Let me first of all start by giving glory to Almighty God for making today's occasion a resounding success. First and foremost, I thank our chief patron, Mr. Ajmal Hossain, SDO Kharagpur, Administrator, Debra Thana, Sheikh Khudiram Siddi Mahavidyalaya. I also express my heartiest thanks to Dr. Rupa Dasgupta, Chairperson and Principal of our college, who, despite of her busy schedule, has found time to grace this occasion. We are highly grateful to our resource persons, Dr. Debra Ghosh, Department of Biomedical Laboratory Science and Management, Vidyashagar University, who, despite of his busy schedule, has found time to grace this webinar. We are also grateful to our another resource persons, Dr. Sukanta Mondal, Principal Scientist, Animal Physiology Division, ICAR, National Institute of Animal Nutrition and Physiology, Bangalore, who, despite of his busy schedule, has found time to grace this webinar. I would like to thank our Joint Organizing Secretary, Mrs. Pallavi Maiti and Mrs. Sridhar Sahu, SAC, Department of Nutrition, Debra Thana, Khudiram Siddi Mahavidyalaya. I also express my thanks to the convener of this webinar, Mr. Amit Kumar Jana, SAC, Debra Thana, Khudiram Siddi Mahavidyalaya. I am also grateful to the joint convener of this webinar, Ms. Swati Bhuya, SAC, Department of Nutrition, Debra Thana, Khudiram Siddi Mahavidyalaya. I would like to thank our advisor committee, Mrs. Bipasha Mojinder De, Coordinator IQAC, Debra Thana, Khudiram Siddi Mahavidyalaya, Mr. Soikat Chakraborty, Joint Convener, Joint Coordinator IQAC, Debra Thana, Khudiram Siddi Mahavidyalaya. Dr. Pankaj Kanti Sarkar, Varsar Teacher Member IQAC, Debra Thana, Khudiram Siddi Mahavidyalaya. Mr. Parthapati Pramanik, TCS Teacher Member IQAC, Debra Thana, Khudiram Siddi Mahavidyalaya. I also express my heartfelt thanks to the enthusiastic technical team, Mr. Somokanti Hota, Assistant Professor in HOD, Department of Department of Mathematics, Debra Thana, Khudiram Siddi Mahavidyalaya. Mr. Subhajit Paul, SACT, Department of Physical Education, Debra Thana, Khudiram Siddi Mahavidyalaya. Mr. Prasanta Dutta, SACT, Department of BCA, Debra Thana, Khudiram Siddi Mahavidyalaya. Without their continuous support, it was not possible to arrange this occasion. Last but not the least, I would like to thank our beloved students, research scholars, teachers, and other audience for making this function as a grand success. Once again, I thank one and all present here. Thank you. Now the mic is over to you, Amit.
Thank you, sir. Thank you all. Now we are going to conclude this webinar. Once again, thank you, our resource persons, and also all our audience. Have a nice day. Thank you.
Thank you.
ঠিক আছে স্যার, ভালো থাকবেন।
Thank you. আচ্ছা।
অমিত স্যার শেষ করলাম।
ঠিক আছে। আচ্ছা।
থ্যাংক ইউ স্যার, থ্যাংক ইউ।
আচ্ছা, থ্যাংক ইউ।